First, I would like to thank the organising committee for awarding me the Beddington Medal. It is a huge privilege and great honour. I would also like to thank my supervisors Bertie Gotkins and Jenny Nichols for their support in providing a wonderful environment which was highly conducive to developing new experimental and computational approaches. My background is that I am a trainee in neonatal and paediatric surgery. Congenital anomalies, particularly structural defects, form the bulk of the work we face as paediatric surgeons. In fact, embryology is the basis for paediatric surgery, so much so that it has been suggested that paediatric surgeons are applied embryologists. Many of our advances in understanding are in fact based on animal models of mammalian development. With advances in technology and increasing amounts of acquired knowledge, integrating large data sets is essentially impossible for individuals, and cross-discipline collaboration combined with computational algorithms becomes essential. This collaboration between multiple labs was key to my PhD work. In complex multicellular organisms, all cells have essentially the same DNA material while producing a wide variety of cell types, all derived from a single progenitor, the fertilized ovum. Characterizing cells and defining their ontogeny is likely key to making progress in understanding congenital defects, but when developing new methods, it makes sense to focus on simpler systems before refining techniques for more complex systems. Additionally, cells may be characterized in several ways, but messenger RNA molecules offer a unique convenience with their poly-A tails allowing capture and readily available methods for amplification and generation of DNA libraries. Additionally, bulk characterization of cells belies any underlying heterogeneity, and rare cell populations may remain hidden and unidentified. Imaging methods have long been able to capture the spatial and single-cell context, but are hindered by the number of assays that can be performed. RNA chemistry allows multiple features to be assayed simultaneously in individual cells, but spatial context is often scrambled. When I joined Bertie's lab, a qPCR dataset from mouse embryos had already been generated. What really attracted me to the lab at the time was the concept of inferring continuous temporal trajectories from course time profiles, commonly known in the field as pseudotime reconstruction. My project itself focused on early murine gastrulation as the mammalian model system and had 4 overarching aims within the context of coarse temporally resolved single-cell data, and they were 1, to define cell populations, 2, to trace lineage trajectories, 3, to simulate developmental processes, and 4, to identify the key molecular pathways. The key findings in my project were the identification and validation of leukotriene induction in early mammalian hematopoiesis, specifically in the differentiation from hemogenic endothelium precursors to erythromyeloid progenitors, where I identified ALOX5 and ALOX5AP to be key players. Additionally, we identified that tau1, a transcription factor key to blood development, is not required to pause early mesodermal cells in an undifferentiated state, as was earlier believed to be the case from in vitro experiments. Computational developments include generating a deep neural network on the qPCR dataset that could faithfully reconstruct in silico gene knockouts known to affect hematopoiesis. I also designed a modification to the TSNE algorithm, now allowing new data points to be added to a pre-existing embedding. This new TSNE method can also allow us to overcome what is the Achilles heel of single-cell data, the loss of spatial context, and it does so by using spatially resolved bulk RNA-seq data to precondition the TSNE landscape and reconstruct spatial context from single-cell datasets. Finally, I look forward to giving a full talk in the near future, but I would again like to thank the organising committee for arranging the e-conference at such short notice.